Right, this is Chapter 12, DNA Technology and Genomics, Part 1. DNA, nowadays we just think it's very obvious that DNA could be used in a courtroom to either solve a case or to prove somebody innocent. That actually did not used to be the case. And so the first person who was put away using DNA is the guy in the picture here. This was in England, and they used DNA not only to put him away, but to exonerate somebody who was innocent of the crime. DNA fingerprinting is a set of laboratory procedures. So it's just not one thing. We're going to do a couple things. It's going to determine with near certainty whether two samples of DNA are from the same individual. And it depends on how much DNA you test. You can be more and more sure the more tests you do, and how rare the genes are that you're testing will again make you more sure. And this now is very common, but even 25 years ago, it was not, it wasn't a given that people would use DNA testing in the courtroom. Okay, plasmids are used to customize bacteria. So when we look at gene cloning, we're going to use a plasmid. And the reason you use plasmids is because they're simple, they're circular, and you can insert a gene right into them. So the way it works is this. If you want to insert a gene into a plasmid, you isolate it. You literally just cut it open. You take the DNA from the eukaryotic cell that you want, the DNA, whatever gene it is you're looking for, and you recombine it into the plasmid. And you look at this word, recombinant DNA, up here, and wow, doesn't that look like a big scary word? All that means is it is recombined DNA. We've taken our plasmid here, we've cut it open here, we took a chunk of a gene out from whatever we wanted from over here, and we recombined it to contain that gene. We stick it in the bacteria, and off it goes, and as it replicates, it will replicate that gene as well. We can then do, one of the reasons we do this is because, of course, a gene is just a way of coding for a protein, and if you want to make a bunch of protein, it's nicer to do that in bacteria than in other animals. They'll just sit there and kick out that protein all day long, and you can use that in, like, for example, insulin's a nice example. You can use that for insulin shots. It used to be that you had to get your insulin from, like, cows or pigs, and now we can have bacteria do that for us, even nicer. So if the recombinant bacteria multiply into a clone, then the foreign genes are also copied. And all that's saying is as they do their binary fission and they go through and they make more and more of them, that foreign DNA stays in that plasmid and gets copied along with everything else. Okay, enzymes are used to cut and paste DNA. And the way that works is you use what's called a restriction enzyme, which can cut DNA at a specific sequence. And then you use DNA ligase, which pastes the new fragment together. So we're going to choose a spot we want to cut the DNA. We have to cut it and then literally paste in a new gene. Okay? So what that looks like looks like this. You're going to have a section of DNA right here that we want to cut. And so this is what's going to be recognized by our restriction enzyme. It's going to come here and it's literally going to cut the DNA. And notice that it doesn't do a straight across cut, right? If you do a straight across cut, if it just cuts it right here straight through, there's no way of pasting then. You don't have what are these called sticky ends. You can't just super glue a piece of DNA there. It doesn't work that way. By leaving this DNA, by cutting it here in a zigzag fashion, it leaves this piece of DNA, and you guys already know that when you find a piece of DNA that has the corresponding pairs here, the A to T and the G to C's, that sticky end here will naturally realign with this sticky end here. So once we've cut it in this zigzag fashion, we leave a sticky end, we take our new bit of DNA, whatever it is we want to insert. It has to be cut with the same restriction enzyme so that you get the same sticky end. So they line up. There's actually different restriction enzymes. We'll leave different kinds of sticky ends. So you need to use the same one. They'll line up beautifully, and bam, you have inserted your gene, whatever that is, an insulin gene, again, whatever that is, you've inserted that into the DNA. And again, here you have the recombinant DNA molecule. That simply means it's recombined. We've combined this with the new gene with the old stuff, all right? We just recombined a set of DNA there. Okay, genes can be cloned in recombinant plasmids. Let's look at it a little closer. Bacteria take in the recombinant plasmids from their surroundings, and they can reproduce, thereby cloning plasmids and genes they carry. Remember, one thing bacteria just do, they just take in DNA from their surroundings. It's a little odd, but yeah, they do that. So we can use that to our advantage. All right, so cloning genes in a bacterial plasmid, and we don't always have to use bacteria, but again, they're kind of nice because they just replicate themselves. This is how it looks. We use E. coli. E. coli is a very common one to use in the lab. Tends to get a bad name, but it actually does a lot of really nice stuff. Yes, it is the same or similar one found in your intestines and the intestines of cow and other things. And if you get the wrong strain of it, yes, it can make you sick. But there's different strains of E. coli. We're looking at the one in the lab, or we're going to take an isolated piece of DNA, essentially a gene. And right here you can see we cut it open. We have the sticky ends. You see how we have these little sticky ends, just like we saw on the last slide. That means we've cut it with the same restriction enzyme. See that little note? You have to use the same restriction enzyme to get the same sticky ends. Otherwise, they will not line up. Then we use DNA ligase just to bond it up. Kind of like super glue, but not quite. And you put the plasmid back into the bacterium. Remember, they will take up plasmids. They do that. And then as it does its normal binary fission, that gene stays with that bacteria. All right, clone genes can be stored in genomic libraries. So we can take all the genes of a human that make protein and put them into a bunch of different bacteria, and we can make whole libraries out of this. A genomic library is sets of DNA fragments containing all of an organism's genes. So, obviously we're interested in human stuff because a lot of these are used for medicine. So maybe we have, the insulin gene, and you have human growth hormone, and you have all these different genes, and they're all in the different bacteria. Okay. It can be constructed and stored in cloned bacterial plasmids or in phages. Either way, remember a phage is just a virus that will infect a bacteria. All right, reverse transcriptase helps make genes for cloning. And reverse transcriptase can be used to make smaller complementary DNA libraries. And cDNA just stands for complementary DNA, containing only the genes that are transcribed by a particular type of cell. So let's look at that a little more closely because that really sums up a lot of information for a pretty cool little thing. Remember when we look at eukaryotic genes, you're going to have introns and exons. And exons are the ones that actually code for the protein. And introns, I like to think of them as interruptons. They interrupt the normal coding. Well, when you go to clone and put these into a bacteria doesn't splice introns and exons. So we got to get rid of those introns first. So we just let the cell do that normally. You have in the nucleus, so here we are, we're in the nucleus here. We need to get rid of these introns, right? So normal transcription, it codes for an RNA sequence. And then we have the splicing, and this happens, and then you make this messenger RNA, which leaves the nucleus. This is just basic transcription and translation, if you remember the transcription part of it. Now we have a piece of RNA that is coded without any introns. So this is what we want. This is just the gene itself. But it's RNA, so we need to make it into DNA. So this reverse transcriptase, and of course I'm sure you can imagine we got this from viruses, which are called retroviruses, which will make DNA from RNA. Remember that whole central dogma rant I do where, we say normally the genetic flow, it goes from a DNA to an RNA to a protein, but retroviruses actually go backwards. They take RNA to DNA. Hence, science can never be dogmatic because it's okay to do things backwards. Um, so this reverse transcriptase will take RNA and make DNA out of it. And because it makes the DNA without the introns, we call this cDNA. It's a complementary strand just of the messenger RNA. So it's all short, it's perfect, it's just the gene we want, it's just going to code for a protein. So no interruptons on that, no introns, no interrupting of that sequence. All right, recombinant cells and organisms can mass produce gene products. What are gene products? Proteins. We're almost always talking proteins here. Proteins, enzymes, proteins. So when we look at recombining, the whole point of this can be to make proteins. When we look at recombinant cells, it can be because we want to mass produce a protein. What kind of things do we want to mass produce? Well, we like to mass produce things called insulin because diabetes is, it's in the millions. I can't remember how many Americans have it, but it's a lot. And the old way of getting the insulin was like from cows and pigs and stuff, and you don't get a whole lot. By doing this recombinant DNA, we can stick that insulin gene into E. coli, and it'll just pump out insulin all day long. Plus, the next beautiful thing about that is that the insulin gene we stuck in there is the human insulin gene, not a cow insulin gene, which is ever so slightly different. Yes, it works, but some people actually had reactions to that. Other things we like, human growth hormone. You have the epidural growth factor that'll help treat burns. So all these things we can produce in a laboratory setting using nothing but bacteria. Now, as you go down the list, you'll notice Saccharomyces cerevisiae. And yes, that is the same Saccharomyces cerevisiae used in beer and winemaking. But, and this is a eukaryotic cell, this is a yeast strand, so some things cannot be made in bacteria. You actually have to make them in a eukaryotic cell. And that's where Saccharomyces cerevisiae will come into play because it can make those, but it is technically eukaryotic. It is not bacteria. And then you see at the very bottom of the list, some things are actually made in mammalian cells. All right, different organisms, including bacteria, yeast, and mammals, can be used for this purpose. So we can use, like I said in the previous slide, you can use Saccharomyces cerevisiae, which is a yeast, E. coli, which is a bacteria, and you can actually use mammalian cells to do some production as well. DNA technology is changing the pharmaceutical industry. And that obviously makes sense, right? Because you used to have to get this insulin like the really hard, complicated way, and now we can just pump it out. It is widely used to produce medicines and to diagnose disease. Both of them are important, and really the advances have been fantastic. Okay. Therapeutic hormones. In 1982, humulin, which is human insulin produced by bacteria, became the first recombinant drug approved for the Food and Drug Administration. So again, this one is the human insulin, not the cow insulin or the pig insulin or whatever they were using before. This one's human, so less side effects, easier to make, and obviously it's been around for a long time. All right, DNA technology can be used in diagnosing diseases. Remember we talked about the genetic diseases, and we talked about, Huntington's and cystic fibrosis, and would you want to know you are a carrier or if you're going to have it if it's Huntington's, and DNA technology can tell you that information. DNA technology is also used to help make vaccines. So when we looked at the things that will be produced, and I said mostly proteins, vaccines and ways for the immune system to help defend against, I think hepatitis is one of these, hepatitis B. So it does more than just make insulin. Okay, nucleic acid probes identify clones carrying specific genes. So when we look at, we're going to run some analysis, some restriction fragment analysis, and we are going to run it on a gel plate, and then we're going to look at it. How do we know which ones are carrying it? Well, you can do these nucleic acid probes, and you can actually make radioactive nucleic acid probes that'll latch on, and that would just be the corresponding piece of DNA. And so it can be used to identify specific pieces of DNA. All right, so let's look at what that looks like. If you have a nucleic acid probe, it's going to be a single-stranded molecule of radioactively labeled or phosphorescent. We like things that glow. We just make it obvious, right? Radioactive. Something that's going to let us know. It's got to be single-stranded because it's a probe. The whole point of this is it's going to go find the other part of the single strand, the bases, and it's going to stick onto that. And literally, when it sticks onto that, you can rinse the rest of this stuff off, and the part that glows will be the part where it's stuck onto a corresponding base pair. So you make one strand radioactive, and you kind of wash the stuff in this, and it will stick onto the correct base pair. Remember, that happens naturally because of the hydrogen bonding. All right, so we do these in what are called microarrays. It's a DNA microarray. It's going to test for the expression of many genes at once. So when you look at doing more than one gene, you do it on what's called a microarray. It's basically a grid that will help you figure out where stuff is. And it can reveal patterns of gene expressions in different kinds of cells. So remember, not all cells are expressing all genes at the exact same time. There are cells where, yes, all the DNA is in every single cell, but not all the DNA is being read in every single cell. And this can be useful because in things like tumor cells, obviously you're going to have different genes being read and expressed than you would in normal cells. All right, so let's look at that little microarray. Remember, I said it's just a grid. We are going to take our messenger RNA. Remember, it's nice. All the messenger RNAs, all the introns and all that stuff, it's all gone. It's just down to what we're looking at. We have the complementary DNA strand made from the messenger RNA. Now we want to know what messenger RNA is being made from these cells. Which proteins are they expressing? So we take this and we put it on a grid, and you can see, the cartoon version, or the actual size is 6,400 genes. So it's a microarray. There's a lot of them. And what will happen is you take these probes and you put it on there, and the ones that fluoresce are the ones that are being expressed. Those are the ones that found the messenger RNA, or the complementary DNA that would be with the messenger RNA, and is actually being expressed in that cell. Okay, so gel electrophoresis sorts DNA molecules by size. So if you chop up DNA, right, just take a restriction enzyme, just chop it up. The big old molecules are not going to go very far. So if you look over here, if we just put in some DNA into these little wells, and DNA is charged, so you can run a charge on it, right? That's going to make it want to run through the gel itself. It's sitting here, right? Well, it's going to get trapped in this gel, and it's not going to move very far if it's a big old chunk of DNA. But if you've cut it into little baby itty bitty pieces, it's going to run pretty far down this gel. So you can see over here, these are the longer molecules. See where it says longer molecules? These are all the big fat pieces. They didn't go very far. And the shorter molecules go farther down the gel. And we can use that information to see what genes are there.